My name is Eric Wice, and I'm a sixth-year PhD student in Julia Saltz's lab at Rice University. And today I'll be discussing how variation in indirect genetic effects shapes and affects variation in social selection. So it should come as no surprise to many of us that the social interactions an individual engages in can have dramatic influences on an individual's fitness. Because social interactions are so important to fitness and a major target of selection, understanding how quantitative genetics shape social traits and how selection acts on this variation is a necessary step toward understanding how social behaviors evolve. To understand how quantitative genetic variation and selection shape social traits, quantitative genetic models have been expanded beyond understanding how natural selection acts on phenotypic variation that's partitioned between genetic and environmental effects, to include the impacts of social partners. When the genes of social partners end up influencing a focal individual's phenotype, these indirect genetic effects, or IGEs, are an additional component of quantitative genetic variation that can shape social traits. Additionally, the presence or actions of this second individual can influence the fitness of our focal individual, and the degree to which it does so is a force called social selection, that depending on the genetic variances and covariances between the interacting individuals can lead to interesting evolutionary outcomes, including the social environment itself evolving. Despite the importance of indirect genetic effects and social selection for understanding social evolution, very little is known about processes that drive variation in IGEs and social selection, which leads me to the main overarching questions I'll be addressing today, which are how variability in the social environment affects variation in both indirect genetic effects and social selection. More specifically, we asked how genotypic variation in social group composition affected the structure of social groups using social network analysis, how genotypic variation in social group composition affects selection on network structure, as well as how a specific type of context-dependent selection, being frequency-dependent selection, varies depending on the genotypic composition of social groups. To address these questions, we need a study system where we can manipulate the genotypic composition of social groups, as well as repeatedly measure the fitness effects of variable genotypes on one another. To do this, we use Drosophila melanogaster flies, which allow us to control the genotypes of individuals using inbred lines of flies derived from a wild population. This also allows us to create replicate social groups of variable genotypic composition, so we can address how the same genotypes have variable indirect genetic effects depending on which other genotype they're paired with. Flies also have easily tractable lifespans of around 70 days, so we can measure multiple components of fitness throughout their entire lives and estimate variability in social selection. Additionally, flies are a great system to work with and address questions about how the genotypic composition of social groups affects structure and selection, as flies have been shown to non-randomly choose and form social groups, as well as form non-random social networks, where flies preferentially associate or disassociate with one another. So to create social groups of variable genotypic composition, we chose 3 heterozygous genotypes of female flies. And then we paired each of these focal genotypes with a group of 9 stimulus individuals of the other genotypes in a full factorial design, but excluding pairings where the same genotype was paired with the same genotype. We then added 10 males of a standardized genotype to each group for a 50-50 sex ratio, and replicated each of these social contexts about 20 times for a total of almost 120 social groups. To quantify network structure, we took videos of social groups and using automated tracking software, created social interaction matrices for each social group. And 2 days after social groups were established, females were removed and placed in individual vials where we measured their lifetime offspring production and lifespan. This allowed us to quantify how individuals affected each other's fitness and social selection. We studied the 5 most commonly estimated parameters of social network position, being in strength, or the number of interactions an individual receives, out strength, or the number of interactions an individual initiates, eigenvector centrality, or how critical an individual is to the overall structure of the network, betweenness centrality, or how much an individual serves as a bridge between indirectly connected individuals in the group, and clustering coefficient, which measures the degree to which the individuals you're connected to are also connected to each other. So it can kind of be considered a measure of how cliquish individuals are. Broadly, we found evidence of direct genetic effects for measures of network position, including eigenvector centrality, betweenness centrality, and clustering coefficient, but not for measures of in-strength and out-strength, which is perhaps surprising given that we might expect an individual's own genotype to have more influence over its direct social interactions compared to measures of network position that incorporate indirect interactions. But this nicely sets up our result for the additional component of quantitative genetic variation we tested, which is how indirect genetic effects shaped individuals' positions within their networks. We similarly found evidence of indirect genetic effects influencing how individuals served as a bridge between indirectly connected individuals, as well as how cliquish individuals were, but not for our other three tested components of network position. So knowing that individuals' positions within their network can be shaped by both direct and indirect genetic effects, we then asked how selection acts on this variation across different social contexts. We didn't find evidence of network position impacting females' lifetime offspring production. We did, however, find selection operating on measures of network positions shown on the x-axis via our fitness component of females' lifespan, shown on the y-axis. And these selection gradients varied depending on indirect genetic effects of which genotype females were paired with. For example, when individuals were paired with genotype B, the yellow genotype, then engaging in more social interactions, either in strength and out strength, or being more central to group structure, eigenvector centrality, had little to maybe a slight increased effect on individuals' lifespan. Yet if individuals were paired with genotype A, the blue genotype, then engaging in more social interactions and being central to group structure were costly for females' lifespan. In addition to estimating selection on network structure, we were also able to test an additional component of context-dependent selection. That is, since the female genotypes in our group were one of either 20 individuals, the focal genotype, or 9 of 20 individuals, the stimulus genotype, we could address how frequency-dependent selection was acting and whether being the rare or common genotype impacted females' fitness. And subsequently, not only did we find evidence of positive frequency-dependent selection, meaning that being part of the more common stimulus genotype, shown on the x-axis, benefited both components of female fitness, shown on the y-axis, we also found that this frequency-dependent selection depended on the social context individuals experienced. For example, being the more common genotype increased how long females lived, but if a female was also paired with genotype A, the blue genotype, this effect was much more pronounced. Typically, positive frequency-dependent selection operates to reduce genetic variation. Less common genotypes are selected out of the population in lieu of more common genotypes. However, because we found that frequency-dependent selection depends on the social context individuals experience, this context-dependent selection can serve to maintain genetic variation. So if we go back to our questions and we set out to address and summarize our results, we find that direct genetic effects and indirect genetic effects shape the structure of social networks. Selection on network structure varies depending on the social context individuals find themselves in. And frequency-dependent selection depends not only on your own frequency in a group, but the genotype and potential phenotypes of individuals you're paired with. Taken together, we can see that the social environment individuals experience can evolve, and perhaps more importantly, how selection acts may depend on the makeup of the group experiencing selection, which is important when we consider that feedbacks between the social environment and selection can shape the evolutionary trajectories of social group structure. And with that, I'd like to thank many people, including my lab mates, all of the students who have assisted me over years, my committee, and numerous funding sources, and I'd be happy to take any questions you may have.